Thank you, John. All right, so I'm really going to be giving a project update on a couple of projects that we have running concurrently. One of them we finished data collection last spring. That's in the St. Clair and Detroit River system. The other is still ongoing. We're collecting the third year of data this spring. So don't let the number of authors fool you. These are very big projects involving lots of co-PIs and collaborators and lots of help. I have changed the title a little bit and added just myself and Mike Lowe here, mainly because Mike has done the lion's share of the work on the St. Clair River data analysis at this point. The preliminary results that I'm going to show and the interpretations are really all ours at this point. They're pretty hot off the press. So lots of other people involved that I'll mention here in a few minutes. And for those of you who aren't familiar with Mike, Mike is a new biologist at Hammond Bay. We've got him for 2 years to lead the data analysis and the writing for the St. Clair River telemetry project. So just real briefly, the motivation for the work that we're doing is this trend here, where the sea lamprey adult index in Lake Erie has been above targets since the mid-90s. And this was particularly surprising that abundance didn't decline after the back-to-back large-scale treatments in Lake Erie, which were thought to have targeted every known larval population in 2008 and 2009. So the work that I'm presenting today really is tied into two sort of really broad hypotheses about why sea lamprey and Lake Erie are still above targets. The first one is that there's an uncontrolled producer out there somewhere that is unknown to the program, possibly a new producer. So if you looked at the historic view of sea lamprey control in Lake Erie tributaries, larval lampricide treatments were focused on about a dozen streams. Some of these are really small and intermittent. And recently, there's been much more attention on the St. Clair and Detroit River system. So this has become, at the time of this work, it was really a question as to what was even there. There's been a lot more work done since. We'll hear some more about this later today, I believe. So attention is really focused on the St. Clair Detroit River system as a potential new producer that's unknown. The second hypothesis is that this sort of trend is sort of an artifact of assessment. This was particularly sort of more concerning when there was only one data point after the back-to-back treatments. And I know Gene Adams has done quite a bit of work on this. And so I'm not going to talk specifically about this, but I'll just walk through real quickly kind of some of the rationale for why one might suspect that there could be an artifact. So first off, there's just a few number of tribs, and this is typical of adult sea lamprey abundance. There's 5 streams in Lake Erie where abundance is estimated. And if we looked at the proportion of that index from that trend plot, we see that it's generally dominated by a couple or a few number of streams. And so it raises the question of whether or not this is sort of sensitive to some of the dynamics that are happening in individual streams. So I'm not going to address this specifically, but the first project that I'm going to talk about in Lake Erie is relevant because we're asking the question, can a lake-wide mark recapture estimator be a useful assessment tool? So this could be in addition to the index. But this is something that sea lamprey, every generation of sea lamprey biologists seems to try to attempt. And this is essentially the idea that you take a batch or batches of marked lampreys, maybe with coded wire tags or other marks, release them into the lake, and then use the trapping data as your recapture data points to estimate a lake-wide abundance. This was last attempted in the 90s by Roger Bergstead. And there were some concerns that essentially the low recovery rates of coded wire tags couldn't be used to really test the assumptions. So now with acoustic telemetry, we can essentially cover all the spawning streams and use, or most of the spawning streams, and use the acoustic receivers in the streams as essentially a recover recovery tool, to increase our recovery rate and hopefully do a better job of testing some of those mark recapture assumptions. So for example, one that I'll be talking about later on is whether or not stream entry and recapture are related to release location. So if we dumped a batch of mark fish in the lake, if whether or not they got recovered or where they got recovered depended on specifically where we put them, then we'd potentially have a biased estimator. So I'll keep moving here. Secondly, one of the questions that I'm addressed today is what fraction of the population in Lake Erie enters and spawns in each tributary. So, essentially looking for streams that sort of aren't on our radar or on the program's radar, where sea lamprey seem to be spawning based on the telemetry data. And the most obvious one here is the St. Clair-Detroit River system. So this question is going to focus on specifically stream selection and using telemetry to identify the proportion of individuals choosing each stream. So for this first study that I'm going to walk through, this is the research team. There's a bunch of other people involved. And so essentially what we're doing here is we put receivers in the tributaries that we believe cover all of the spawning habitat. At least these are the streams that have had larval production, and then some extras like the Maumee, for example, simply because it's a big river in the western basin, and we were curious if maybe there were sea lampreys going up those streams. What we attempted to do here was to put receivers into every tributary that has been known to recently have larval sea lampreys. So based on the data we have, we think this encompasses all of the adult spawning locations. The stars are the release locations, so we distributed them around the lake, and essentially what we're looking at is whether or not where we release influences where they go. The basic approach here, I'm not going to dwell on this too much, is that you have a number of release locations, and from any one of those, the fish can choose to enter any tributary. So we have some parameters that we can estimate that represent the proportion of the fish from each release that selected each stream. If you sum those up, assuming that we have all of the habitat covered, and that's an assumption we'll have to think about more carefully, but if you make that assumption, then you have an estimate of survival. You can sum all those up and say if they entered a stream, they survived to stream entry. Otherwise, there's some fish you don't detect, and you might consider that to be a mortality, the proportion that died. More importantly, we can sort of factor out maybe release-specific mortality by dividing the proportion that went to each stream by survival, and then we sort of have the proportion of the spawning population or the adult population in each stream. So really what we're interested in is comparing these transition probabilities, the stream selection probabilities, for each stream among releases to test the assumption of a mark recapture estimator. So the key proportions are the survival probabilities and the stream selection probabilities. This is sort of our plan for the model. We haven't gotten there yet, so I'm going to show basically some descriptive results. But some other extensions of this are potentially, depending on the data, is you can have lampreys, once they're in a stream, either remaining in a stream or getting trapped, so essentially estimating trap efficiencies within streams, or going back to the lake, and this is essentially a rejection rate. And so some lampreys, even though they enter a stream, may not spawn there. They might leave and go to another stream and repeat the cycle. So other parameters that are sort of on our radar that I'm not going to dwell on today. All right, so I'm going to real quickly just walk through some descriptive plots just to kind of show you at this point sort of what's happening. Not at the point where we have some strong, clear take-home conclusions, but I think there's some broad patterns here that are worth presenting. So, for example, we released adult sea lampreys in 2016 in the north central basin, and the symbols, they're hard to see because they're small, but the color on the symbols is reflective of the number of fish from that release group that entered each of those tributaries, were detected on those receivers. So at this release site, we released 30 in 2016 and 23 in 2017. We detected 91% of those somewhere later that year, and some number of those were trapped. These are on the slide, but I'm not going to even talk about them as I go through these. So I'm going to quickly just walk through each of these for each year. I'm not going to get into the details, but I'm just kind of going to try to point out that north central release in 2016, those lampreys are showing up in the eastern basin, not so much on the U.S. side in the central basin, and some of them showing up in the Detroit River. 2017, by and large, a similar pattern. A few more showing up on the southern side, on the opposite side of the central basin. So right away, this is telling us that it looks like, based on this one release group, there may be a tendency for them to stay close to home or close to release, not home, but they're also pretty wide-ranging. If we move to a release in the eastern basin, we see a similar kind of trend. Maybe not so much movement across the entire lake. But again, these are released as adults. So, there was a concern that the adults might just be able to find the nearest stream and ascend it. And clearly, we're seeing that at least they have a tendency to explore the entire lake. They're finding these streams within a week or two of release. So generally, what we're finding is that they're pretty wide-ranging. Here's a release in another one on the U.S. side in the Central Basin, where we tend to see them ascending rivers nearest the release at the highest rate, but again, widespread distributions. There's a release also in the Central Basin. Start to see some of these in the Maumee even. And in this case, the Detroit River seems to be seeing just as many lampreys as some of these other streams. So I'm just going to finish flicking through these real quick because I think the point has been made. And we'll talk about now juveniles. So these are released in December and January before they're going to spawn. So at the beginning of the winter before they spawn, these are at maximum size. And this is a different kind of plot. I'm actually just going to show numbers here instead of the shading. These one of the issues we had with juveniles was just getting lampreys to release at this time of year. So these were record low catches for the past decade, and it happens to coincide with our project, unfortunately. We were expecting hundreds or thousands based on the historic data. But we released what we had, and then we put the rest of the tags into adults. So at any rate, we detected 29 of them. So this is overwinter survival, essentially. I mean, we're going to put error bars on this when we actually do the statistical analysis. But for now, as a preliminary assessment, more of these are spread out across all releases. The sample size is so low that I'm not going to bother showing individual releases. But more showed up in the Detroit River than any other tributary. Next was Big Otter Creek, one of those trap sites. 7 lampreys there. 4 lampreys at Big Creek, 4 at the Grand River in Ohio, so those are both trap sites. 3 in the Maumee, which is a bit of a surprise. 2 in the Grand River in Ontario, which is not a trapping site. 2 in the Cattaraugus. So you can see that the data are sparse and the numbers are small. Unfortunately, this is sort of pilot level. But again, I think the take home here is that good thing for the program, those 5, at least 4 of the 5 trapping rivers seem to be getting most of the lamprey. But we're also seeing a bit of a surprise with the Maumee and maybe the Grand, and then the Detroit seeing more of those lampreys than any other. Only one of them was caught, so about a 3% capture rate, which seems low, but again, I'm not going to dwell on that today. And that was caught in Little Otter Creek. So going on to 2017, we got 49 lampreys. Again, a really small sample size. We were hoping for over 100. Proportion detected was a little lower. This time, Big Creek had 6 of them. The Maumee also had 6. Cattaraugus had 5. Grand River had 5. Big Otter Creek had 4. So again, those 4 rivers with traps seem to be seeing most of the lamprey, and the Detroit River kind of falls up with them as well. So I'm going to move through this. Real surprise here was that the Credit River in Lake Ontario actually detected 3 of these lampreys. And this is thanks to Sarah LaRoque for passing this information along. We suspect that we had some other data from one other lamprey in another year that had actually passed all the way through the Niagara River. So now this raises some real questions about whether or not we're really capturing survival like we thought we were. If 3 of these lampreys, this is on the order of what we're seeing in some of these Lake Erie tributaries, especially the big producing Lake Erie tributaries, 3 of them are able to pass through the Niagara River or the Welland Canal, get into this one river in Lake Ontario. It really makes us wonder how many other lampreys from Lake Erie are moving and contributing to Lake Ontario's population. So thanks to Dmitry Gorski and John Midwood, we're going to have receivers in the Welland and the Niagara that can detect these tags coming up. So preliminary conclusions from this Lake Erie work. Number one, I think it's safe to say, again, it's too early to make any real hard conclusions relative to our specific objectives, but the adults were generally wide-ranging. I think that's safe to say. But there is some preference for tribs in the basin of release. So our next step is to try to understand, based on those transition probabilities, those stream selection probabilities, if we did a single release mark recapture estimate, how strong would that local stream selection tendency be, and how might that affect an adult population estimate at the lake-wide? We also see some basin-specific differences, so similar. We also see pretty consistent use of the St. Clair-Detroit River system. And this is not unexpected, but this also raises the next question of how important is understanding the adult population size in that system. So I'll transition now to the study that we had in the St. Clair-Detroit system. I think it's safe to say now that we know pretty well that larvae are distributed throughout the St. Clair River in particular. But we don't know what proportion of those contribute to Lake Erie. The Commission has funded some work that Nick Johnson and others have started to try to understand this by tagging and recovering larvae. But we also don't know much about adult abundance. Adult abundance might be important to understanding it might have implications for the index at the lake level, but it also would be very important for something like sterile male releases if those were used to control the St. Clair. Finally, we don't really know where they spawn, and we don't know if there are hot spots, if they're spawning in tributaries versus the main stem, and there are no traps in this system in the St. Clair or Detroit rivers or their tributaries for which to estimate abundance. So understanding their spawning migration routes and locations of spawning might lead us to develop some ways of estimating adult abundance. So what's on the table? As far as I know, the Commission is still considering treatment with balicide. The cost would be astronomical, but probably the only option on the table at this point. Sterile male releases are also an option, but this would be heavily dependent on some understanding of adult abundance, so you'd know what the ratio or how many sterile males you'd need to release. So again, needing to understand more information about adult sea lamprey in the system. So this second study, we collected 3 years of data. The 2 years just concluded. Mike Lowe has taken the lead, but here are the other members of the research team. We released 27 lampreys as a pilot study in 2014 at the mouth of the Detroit River, and then 125 and 145 in 16 and 17. Today I'm going to show preliminary results from 2016. The receiver locations are these dark blue symbols. They're basically throughout the whole system, and then in the 5 tributaries where lamprey have been seen in the past. I'm going to show essentially a series of plots with color coding where Mike has divided the river up into polygons, and this is just his first cut at this analysis, so this is intended to be pretty coarse scale. But essentially we've got color coding for the lower Detroit, the upper Detroit, the Lake St. Clair, where we don't expect there to be any spawning habitat, the lower St. Clair, middle St. Clair, upper St. Clair, and Lake Huron. So he's using, at the moment, he's using sort of a sequence analysis and cluster analysis. And so he's taking the history of each lamprey, starting when it's released, and on each day he plots the location of that lamprey as a sequence. So this is a lamprey that would have moved up the river within a few days, maybe poked its head into Lake St. Clair, and then moved back into the Detroit River. Here's one that would have moved through Lake St. Clair into the lower St. Clair and then stayed in the middle St. Clair. Similarly, fish that moved up, lamprey that moved up into the middle St. Clair and then back into the lower St. Clair. And then finally, one that moves all the way through the system into Lake Huron. So here's a quick animation that just illustrates sort of the characteristic fish, where now you can see after a week or two, we have a fish in the Detroit, one that's up in Lake Huron, and then we've got a couple here in the middle St. Clair, and one of those will fall back. So that just sort of illustrates the sort of characteristics of four of the major groups. So what he's done is he's taken those sequences, he's stacked them up. So this is looking over time and days since release. Each fish is on the y-axis with that movement sequence that I displayed. And then he's using cluster analysis to group these into, at this point, the analysis identified 7 groups, predominantly sorted by where they end up. So, for instance, 2 of the fish never really left the lower Detroit River or Lake Erie polygon. 12% or 15 remained in the actually this is the upper Detroit River. 14% Lake St. Clair, 28%, so on and so forth. And so this is sort of our population sort of picture of where these lampreys ceased migration. At the simplest level, you might assume that this is where they spawned. But if you note, there's also a lot of stuff happening back in here where fish are switching states. So there's actually some much more data contained in these sequences about the fish behavior that we haven't even talked about. I'm going to try to go through this really quickly for the sake of time. But just to remind again, there's this group of fish now that we're looking at where 20% of the fish that we release, so we assume these are representative of the spawning population in this system, ended up in Lake Huron. And so I'm going to show these time series plots where now we're just looking at that group. The formatting is a little goofy when I switched it from widescreen to standard, but that's okay. This is along the y-axis, the proportion of this group that's in each location state on each day of the year. So essentially what's happening is early on, you're seeing fish moving up and passing into Lake Huron. Then there's a little bit of this lag, and then there's more fish from the lower river, particularly from the middle St. Clair, moving up into Lake Huron. So there's a lot of information about the fish's behavior that's important to adult assessment even, but just to real quickly show a more complex case. Now we're looking at the fish that ceased migration in the lower St. Clair, and it seems that there may be 4 sort of behavioral stages that are evident in this plot. The first one, what we're seeing is in the beginning, again, fish moving upstream through the Detroit and Lake St. Clair. And about 2 weeks after release, we have a small proportion in the upper St. Clair, most of them in the middle St. Clair, and some in the lower St. Clair. Then we have about a 2-week period. So this is, you might consider to be active upstream migration. Then we have this 2-week period where things are pretty stable. Fish aren't moving around much. They're holding station, maybe waiting for spawning conditions, maybe even building nests. We don't know. But there's this sort of period of holding, whether that's resting or staging or what. But then there's about a 2-week period where there's more active migration, but in the opposite direction. Active movement from the middle St. Clair and the upper, but mostly the middle to the lower. And this is that fallback behavior. And so in understanding where and when these lamprey are spawning in the system now, we're trying to untangle what is happening here, if they're spawning and then falling back, or if they're falling back and then spawning, and that has implications for where we assume spawning is occurring. And just as a final example, we see something very similar for fish that ultimately end up in Lake St. Clair, where we don't think there's any spawning habitat. Some dropping back from upstream, but also some moving up from downstream. So back to kind of the big question, where are they spawning? Well, consistent with our pilot study, the 2016 data anyway indicate that primarily it looks like the St. Clair River is the main spawning location in the HEC. This is consistent with the larval assessments, which is good. It's also consistent with the pilot study. And importantly, there's no evidence of use of tributaries. So we were hopeful that there would be enough fish going into a tributary that maybe you could put a trap in there and get some kind of abundance estimate for the system. Doesn't look like that's the case, but maybe this opens the door to using pheromones or some other method to try to draw lampreys in there, either to kill them or to count them. We were a bit surprised in 2016 because we saw more in the Detroit River than we expected based on the pilot. This could just be because we had a larger sample size. We don't know. Maybe conditions were different. Maybe 2017 will help. And then we saw a lot more move into Lake Huron, which was a bit of a surprise based on the pilot results as well. So last thing I'll show real quickly is what I guess maybe I got cut off. To try to understand the finer scale distribution in the St. Clair River, we used receivers on these drifters, and then we just floated them down the river and kept an eye on them. And we were hoping, again, that there might be hot spots of lamprey distributions that could then be utilized for some kind of market capture enumeration effort. And that, unfortunately, was not the case. We see widespread distributions throughout the St. Clair, particularly in the middle St. Clair. So that's not necessarily good news for the program, but good information going forward. So I'm going to stop there, and there's obviously a lot more work to do, but this is my first presentation with this group, and so I'm hoping that this will, spin up some useful discussions